everyone, today we're going to be talking about different types of chromatography. Ion exchange chromatography purifies proteins, peptides, amino acids, and nucleic acids, and separates them based on their net surface charge. When the column is loaded with a particular molecule with a given pH, all the molecules with a charge opposite to the column resin will bind to it and elute. Size exclusion chromatography separates molecules based on differences in size as they pass through resin packed in a column. The gel consists of beads containing pores of a specific size, allowing smaller molecules to enter the beads while excluding the larger ones. Affinity chromatography involves the manipulation of protein-ligand interactions such as enzyme and substrate to purify a specific molecule or a group of molecules. These reversible interactions are done by placing affinity ligand onto an insoluble matrix to make up the stationary phase and having the target molecule in the mobile phase. The differences in the three types of chromatography is in their nature of interactions and how they separate the molecules. First, in affinity, the ligand reversibly binds to the target molecule. In size exclusion, it separates based on size, and in ion exchange, oppositely charged ions interact to separate the ion from the mixture. What type of chromatography is typically used to purify a protein with a glutathione S-transferase tag? Typically, affinity chromatography is used for this process because it allows the manipulation of enzyme-substrate interactions. There are 3 steps involved in the purification of a GST-tagged protein.
Step 1. Binding. Glutathione is a tripeptide consisting of glycine, cysteine, glycine, amino acids, and is a specific binding substrate for the GST enzyme. A near-neutral buffer, such as a tris buffer with a pH around 7.5, is used to create effective binding conditions. The buffer is first added to the column and allowed to flow through, followed by the GST-tagged protein, which is also flowed through to allow unbound proteins to flow out of the column.
Step 2. Washing. The bound proteins are washed off the column. The same near-neutral buffer is added again and then allowed to flow through. This step is typically repeated 4 times for a total of 5 washes to allow unbound components to be washed off the column.
Step 3. Elution. The purified GST-tagged protein can be dissociated off the column and eluted by the addition of an elution buffer containing excess reduced glutathione to the column. The free glutathione competes for bound glutathione as a substrate for GST's active site, which allows for the displacement of the immobilized GST with the excess glutathione, resulting in elution of the GST-tagged protein in the flow-through. The flow-through is collected in a microcentrifuge tube and is ready for STS phase analysis.
In this video, we talked about 3 types of chromatography: ion exchange, size exclusion, and affinity chromatography. An example that we discussed for affinity chromatography is the purification of GST-tagged protein.